I welcome you all once again. I am Namrata Ashok from Dean ACR DRF team, IIT Bombay, and I will be your host for today's event. The 10x GMP facility at IIT Bombay is the first of its kind and will allow for seamless transition from concept to commercialization. By fostering collaboration, innovation, and excellence, the facility will redefine medical solutions, drive progress, and accelerate advancements in the healthcare and biotechnology. While the 10x GMP facility currently serves the biotech and healthcare domains, it is built for the broader applications in the future. I request Professor Balaji, Head of Department, Biosciences and Bioengineering, to deliver the welcome address. May I have you on the stage, sir? Thank you.
Good morning, Professor Subhashish Chaudhary, Director IIT Bombay, Dr. C.S. Pramesh, Chief Guest for today's event, and Director Tata Memorial Hospital, Padma Shri Dr. Raman Gangakidkar, Guest of Honor for today's event, and Distinguished Professor at Symbiosis International, a deemed university, and formerly with ICMR in various different roles, Mr. Raj Nair, donor for the setting up of this 10x GMP facility, also a distinguished alumnus of IIT Bombay, Dr. Gopi Bhubaneswar, guest speaker for today's event, and an independent advisor and technical consultant in medical device design, development, testing, and quality management system, Professor Krishna Rao and Professor Sudarshan, Deputy Directors of IIT Bombay, Professor Ravindra Gudi, Dean, Alumni and Corporate Relation, as well as other Deans and Associate Deans of our institute, faculty colleagues, both former and erstwhile, alumni of IIT Bombay, industry partners of IIT Bombay, other dignitaries, and members of the IIT Bombay community. I welcome you all to this inauguration event. I will tell something that I repeat almost like a mantra in every such event. I am actually very proud of our department, since out of nearly 30 faculty members, nearly half, 15, are in an entrepreneurship role. I would not have imagined that a department of biology in this campus would have come this far, because about 24 years ago, people used to ask, what is biology doing in IIT campus? This is an engineering institute. And this has been a phenomenal transformation, not just doing fundamental research, but also doing applied work. We are into various stages of commercialization, technology transfer, founding new startups, as well as advising startups, all the different parts related to entrepreneurship. The foremost among this is Professor Rahul Purwar, who gave to this country the indigenously developed Made in India CAR T-cell therapy, and that was done in collaboration with clinicians from Tata Memorial Center. And in fact, this was dedicated to the nation by the Honorable President of India, Ms. Draupadi Murmu, from the same venue just about 2 to 3 weeks ago. And it is one of the finest examples of Made in India and Atmanirbhar India. This is really a crowning glory for all of us. In addition to that, we have so many other technologies that we have, point of care devices, many are there that have been developed in our department and commercialization various stages. We have also developed a small animal perfusion imaging system, We have set up company to provide state-of-the-art genomic solution towards personalized medicine. About 150 head count is of that company. And we have developed point-of-care technology for screening of sickle cell anemia. It's in various, some stage where the commercialization, the discussions are going on now. We have technology for increasing the nutritional value and per acreage yield of important crops, specifically the potato, which has already been field tested and ready. We also have tissue engineered scaffolds for drug delivery and treatment of burn injuries and diabetic foot wounds. We have technologies for early detection of Parkinson's disease. We have technology for identification of biomarkers for various types of cancers. Whatever I'm telling, they're all in different stages of a company, either completely commercialized, completely technology transferred. And we also have a metabolically engineered type strain for bioremediation. It is a type strain, completely indigenous, and many others are there. So what I see is that this setting up of the 10x GMP facility in our department is not just an accidental. It is actually going to make a big difference for the technology development effort and really give big boost. Right now we have 3 technologists, 2 from our department and 1 from chemical engineering department by Professor Jayesh Ballare. And you will get to hear these things later in the day. So overall, as head of the Department of Bioscience Bioengineering, I am extremely happy that this facility has been set up. One of the remarkable things is that this whole thing was done in about a year's time, and Mr. Raj Nair not just gave the monetary donation, but he gave so much time. Almost every week he used to have meetings to make sure that things are on target, and Mr. Ajit Javle also contributed a lot in setting up. This is really remarkable, and I really mean it. So with these brief remarks, I welcome you all once again to this inauguration event. Thank you.
Thank you, Professor Balaji, for your address. Now we have a video message from Dr. Sharad Shroff, Chairperson, Board of Governors, IIT Bombay.
Chairperson of IIT Bombay, Board of Governors, I am honored to address you virtually to mark the inauguration of GMP facility at IIT Bombay. I am sorry I am not able to be there in person as I am traveling overseas. This facility is made possible by the visionary generosity of our dear friend Mr. Raj Nair and stands as a testament to the enduring bond between philanthropy and progress. Through this facility, IIT Bombay is poised to revolutionize the translational research, bridging the gap between academic inquiry and real-world impact. From breakthrough nanotechnologies to life-saving medical advancement, the 10x GMP facility embodies our commitment to innovation with a conscience. I once again would like to thank my friend Raj for bringing this legacy at his alma mater. I am sure the GMP facility will serve as a fountainhead of impactful healthcare products, and I wish the function all the success. Thank you very much.
Thank you, Dr. Shroff, for sharing your insight. Now I invite Professor Shubhasish Chaudhary, Director, IIT Bombay, to address the audience. May I have you on stage, sir? Thank you.
Good morning. Dr. C.S. Pramesh, the chief guest of the program today. We have Mr. Raj Nair and his entire family here. Then we also have another very prominent alumnus who has played a stellar role in getting this particular program done, Mr. Ajit Javle, Dr. Raman Gangakhedkar, other distinguished guests, faculty colleagues, students, and alumni. It's a great pleasure for us to see that today we are doing something very different. Maybe 10 years ago, if somebody had said that we are setting up a GMP facility in a technology institute where biology is just one aspect of that bioscience. But as Professor Balaji just now mentioned, the remarkable kind of progress that this department has achieved over the years, culminating into so much of developments, so many patents, okay, so many other things which are almost ready in the market, including what was inaugurated by none other than the President of India, that is the CAR T-cell therapy. That is testament to what IIT Bombay can do on a specific area for which I think earlier people said, no, this is completely beyond the scope of IITs. Okay? Not only IIT Bombay, but probably all IITs. But days have changed, and so are the ways the research is shaping up in places like IITs, and IIT Bombay is definitely one of them, which is trying to keep pace with the needs of the people of the country and at large. What today, I remember distinctly the day that I met our distinguished alumnus, Raj Nair, that time. He had a horrid time, I know, okay, at a personal level, but he wanted to do something, okay, and wanted to do something that would get us a big boost in certain things, the facility which IIT Bombay did not have. And when he suggested that it is GMP, of course, that was the first time I even heard the name. There is something called, GMP. So I had to look up Wikipedia kind of thing to see what it is. But I think we really realized that the need for that, particularly when we talked to some of our colleagues in the biosciences department, and we realized that yes, need for that is now, and we must get into that. But at the same time, okay, probably we are not ready in the form of what it takes to build such a particular kind of facility for IIT Bombay. And it was a lot of learning experience for all people. Of course, I had a very little role there, but I think that the people in the department, I think Professor Balaji would probably attest to what I'm saying, that learning how to actually build this GMP, which is a very, very critical kind of facility that is required, because we are talking about healthcare where absolute kind of, I would say the precision as well as the quality that goes in is very important. So it was not the philanthropy that I would emphasize here, but it was his engagement with IIT Bombay. So he realized that without external help, we cannot probably complete this. And then we found another wonderful person, Mr. Javle. He's another our alumnus, and I think it has been like almost every weekend there were meetings, how to do it, how to circumvent the problems. And of course, as you all know, anything you build, before anything happens, there is something called BMC, which, okay, is of course slows down things, okay. And thankfully, we could even get out of that, and it is all today, it's all done, and it is ready to be, put into use. So we are so thankful to Mr. Raj Nair. We are also so thankful to Mr. Javle for their so much of engagement with us. But I would also say that it is not just for only the research. The research is, of course, at the first point that we must do. We have large number of PhD students. Our faculty members are very much involved. But what it also requires is that it should, whatever the output of the research should get translated into product so that finally it goes into the market. It helps the common people. Healthcare is one of the most needed thing in the country, and we must ensure that, our research output gets into the product. And for that, IIT Bombay has, again, with the help of all our alumni and many other such kind of, people, we have set up the Translation Research Center. And I'm happy to say that, we have, yesterday only, we set up a specific vertical under the Translation Research Center. It is on membrane technology, which has probably a lot of connection with what we are doing. Today in this, some of the people are common, so, at least I could see Professor Jayes Vallari smiling, so he would be using both of them. And this is something that we want to see that, again, of course, that membrane, the hub that we actually got set up under the Translation Research Center is, again, thanks to, again, alumni, philanthropy, okay, to set it up. But the reason that I'm saying is that we should make sure that the research that comes out goes through that particular, it doesn't stop right at being publications or patent, but gets into translation, because many of them are extremely useful thing for the society, so that should happen. And if that happens, if you go through the Translation Research Center, whose purpose is to boost up the TRL level, okay, and then from there, we have just right next door the incubator, okay, that is SIGN, Society for Innovation and Entrepreneurship. So I am sure from there we would like to see a lot of such things going into the SIGN as an incubation, or, and after they graduate from SIGN, we have a proposal for that also. So we have on the other side of the pipeline, the research park, which was dedicated by the Prime Minister, okay, just about, 6 weeks ago, okay. And so there we would like to see some of these companies and others which are, actually being placed there and working with hands-on with our people there for the benefit of all. So I don't want to take up any more time. It's been a wonderful, I think, initiative initiated by our distinguished alumni. And we would like to promise to them that on behalf of the Institute, on behalf of my colleagues at the, they're here in biosciences, that we'll try our best to ensure the best use of that. Thank you so much, and we hope that it will be a wonderful experience for us.
Thank you, Professor Chaudhary, for addressing the audience. Now I invite Mr. Raj Nair and other dignitaries for the virtual inauguration of the 10X GMP facility. May I have you all on the stage, please?
Please. You can come down now? Yes. Okay, thank you. Do you want to sit there? Yeah, I will.
Now get ready for a virtual walkthrough of the 10X GMP facility, which marks a significant milestone in our commitment to bridging the gap between laboratory discoveries and real-world solution.
Introducing the new 10x Good Manufacturing Practices facility at IIT Bombay. This state-of-the-art facility bridges the gap between research and solutions that are ready for clinical trials. 10x GMP will provide a production facility to researchers who have solutions which are 10 times cheaper, better, faster than available remedies. This facility has two distinct parts. The sterile portion, where final stages of the product manufacture and filling is done, and the preparatory portion, where initial and pre-final stage of the product development is carried out. The entry of researchers into the preparatory portion of the facility is through a series of chambers with a predetermined clothing change procedure to achieve the required level of hygiene necessary for the preparatory portion. In the prep area 1, presently initial process steps related to bacterial, DNA, plasmid projects are planned to be carried out. In Prep Area 2, unit process operations like media preparation, etc. are carried out. This clean corridor facilitates movement of personnel and material between the preparation areas and other support areas like material dispensing, washing, etc. Here in the washing and sterilization area, washing, drying, sterilization operations are carried out using equipment like autoclave and dry heating system to ensure the sterility of materials and equipment used in manufacturing process. Once sterilized, materials are transferred from the designated equipment through a sterilized corridor to the dedicated sterile suites through dynamic pass boxes to guarantee continued sterility. Maintaining sterility is paramount. After placing the materials and accessories in the pass box, researchers follow designated exit routes. The researcher enters a sterile suite with an appropriate change procedure, which is more stringent as compared to the prep area entry. The sterile suites are the key process areas where the final steps of product manufacture and filling or sealing are completed. Presently, Sterile Suite 1 is planned for vector and cellular projects and Sterile Suite 2 for nanotech projects. This state-of-the-art 10x GMP facility will drive faster translations of transformative research advancements into tangible healthcare solutions. Currently, the facility focuses on high-quality projects spanning nanotechnology, vectors, cellular research, bacterial plasmids, and DNA. It enables concurrent development of multiple projects and products, facilitating swift progress. The in-house production capabilities ensure compliance with regulatory requirements for clinical trial batches. Moreover, the facility is committed to bolstering critical public health initiatives. This on-campus facility empowers IIT Bombay to utilize its resources for efficient commercialization, ultimately leading to faster delivery of life-saving solutions at reduced costs. The 10x GMP at IIT Bombay, a testament to innovation.
This state-of-the-art 10x GMP facility will help drive transformative advancement in the development and translation of healthcare products. I request our chief guest, Dr. C.S. Pramesh, Director, Tata Memorial Hospital, and the Professor and Head of Thoracic Surgery at the Tata Memorial Center, Mumbai. He serves on the UICC Board of Directors. He is the convener for the National Cancer Grid, a large network of 325 cancer centers in India. He is also a visiting professor at the Division of Cancer Studies, King's College London, and the Institute of Cancer Policy, King's Health Partners, London. May I request Dr. C.S. Ramesh to be on stage. Thank you, sir.
Good morning, ladies and gentlemen, and dignitaries who blessed this occasion. At the outset, let me thank Professor Goody, Professor Balaji, and Raj for the honor of being able to participate in this momentous occasion. I say this is momentous because this bridges a gap which we've had for several years in academia. If you look at most biomedical advances globally, a lot of the initial development right up to the stage of human trials happen in academic centers, unlike in India, where much of the discovery part of research actually happens in the pharmaceutical industry. And I'm hopeful that this GMP facility will be something that changes this entirely on its head. So most of us who do research go through this valley of death, where from a research idea to a concept to a prototype, right up to the time it gets you a publication in a paper, is done very well by most researchers in India. Unfortunately, the translation of that into a product which is important for society is not as much as it should have been. We had a stellar example of how this valley of death was overcome a couple of weeks back when the team at IIT Bombay, led by Rahul, along with the Tata Memorial Center hematolymphoid group, actually came out with a solution which comes at exactly the same quality as what is available abroad, but at a cost which is a tenth of what is available. And one thing I would really like to emphasize, and that's something that's getting lost in a lot of the Make in India, Atmanirbhar fervor that we have, is that while we strive for cheaper, less expensive innovations, let us not forget that this cannot happen without the high stringent quality measures that are being involved. It has to be high quality with a cost-effective intervention. And I'm really glad that the 10x GMP facility, I really like the name 10x, the fact that it's not just 10 times cheaper, but it's also 10 times better. And I think that's an important aspect that we need to focus upon when we go ahead with medical progress. I must congratulate Raj for his generosity, as well as the IIT Bombay leadership for its visionary initiative of making sure that something that is so unlikely in a technology institution is now will probably serve the population of biomedical researchers across the country. And I have no doubts that this will be a facility that will be heavily in demand amongst medical researchers across not just Bombay, but across the country as well. The importance of coming up with our own solutions is very, very important. Let me talk about some of the progress that we made in cancer research. If you look at global data, we've had improvements in cancer survival, especially in the last 3 decades. Since 1990 onwards, we've had a steady improvement in cancer survival with practically all the cancers that we study. When we look at mortality, when it's age-adjusted mortality, there's been almost a 1% reduction in age-adjusted mortality every year. So from 1990 till today, we have a 33% reduction in mortality, which is nothing short of stunning. But I must qualify this with some other data. Age-standardized mortality is one aspect where we can celebrate the progress of biomedical research, but when you're looking at it from a policymaker's perspective, you need to look at the absolute numbers. And if you were not to adjust for age, there has actually been a 40% increase in cancer mortality. And if you were not to adjust for the increased population, you actually have an 80% increase in cancer mortality in just 30 years. So while we might celebrate the progress that biomedical research has made in getting age-standardized mortality down, what a policymaker needs to think about is the absolute number of patients diagnosed with cancer and their outcomes, and that has been an 80% increase. So clearly a manifestation of something that's very good, which is that people are living longer, and therefore more people get cancer, and the fact that the population has been increasing has resulted in this. But nevertheless, the burden that we are faced with is actually increased substantially. When we look at some of the products, and here I'll use drugs as an example, there's a study that looked at 71 consecutive drugs approved by the US FDA, and what was looked at was the median improvement in survival. Contrary to what we might believe or what we might like to believe, the median improvement in overall survival with 71 consecutive drugs approved by the US FDA was just 2 months. And this has come at a cost. This has come at a substantial cost. If you were to look at the same 30 to 40 years progress that we are looking at, the average cost in the United States for cancer treatment has gone up from an average of $100 per month to $10,000 per month in just 3 decades. And now, probably most societies, including high-income societies, will find this extremely difficult to sustain. And I think even the United States healthcare system is going through a major crisis of its own. We did a study on 700 consecutive cancer trials, which were potentially practice-changing, published in some of the major journals. The data is quite sobering. 92% of these 700 randomized trials which changed practice were led out of high-income countries. 87% of them studied chemotherapy drugs, which is systemic therapy. So a total of 8% were the trials run out of low and middle-income countries, and just 13% was research done on surgery, radiation, palliative care, survivorship, which contributes so much to overall cancer control. So clearly there's a huge skew in the way cancer research is currently being done. The final example I want to give about the skew is about the burden versus the investment in cancer research. Lung cancer, which I treat, is responsible for 20% of all cancer deaths globally. It gets 7% of the overall cancer research investment. And there are examples which go the other way, with breast cancer, which is responsible for 7% of the overall cancer mortality, getting 18% of the overall funding. So I'm not for a moment suggesting that we need less investment in breast cancer research, but what we need is a more neutral and burden-related investment in research. Which is why when we realize that 70% of all cancer patients diagnosed with cancer will come out of low middle income countries, I think it's time that we drive the agenda on cancer research across the globe and not allow only 8% of the research to be done from low and middle income countries. Working with a bunch of really smart global cancer experts, we identified 5 major priorities that cancer research should have, and we published this recently in Nature a year or 2 back. And the priorities that we identified were the following. The first, to reduce the proportion of advanced cancers being diagnosed in low and middle income countries. The second, to improve access and affordability of cancer care. The third, to look at health economics and health technology assessment to drive policy in cancer management. The fourth was to leverage technology in an effort to leapfrog some of the advances that we've seen in high-income countries in a much shorter time. And finally, looking at implementation research and quality improvement as some of the drivers for overall cancer control. So the reason I mentioned these 5 is that none of the high-income countries prioritize these 5 important areas for progress in cancer research. It's primarily driven by the pharmaceutical industry, which is okay. Drugs are one part of the entire spectrum, but ignoring all the other areas of research, including implementation science, health economics, technology, would be extremely foolish. So clearly, I think it's time for us to step up to what we need to do in cancer research. We also looked at identifying what were the challenges facing biomedical researchers in India, and it came down to 3 or 4 major areas. The first was the lack of training. In undergraduate and postgraduate medical education, though theses are part of the overall progress of a medical student, these are really not trained in the way that they should be to make them future researchers. The second is the lack of protected time. With the busy clinical workload that individuals have in hospitals, research is far from their minds when they embark even on an academic career. Funding specifically for clinical forms of research is something that is extremely difficult to get in India. The Department of Biotechnology and the Department of Science and Technology focus predominantly on basic science. And the Indian Council of Medical Research, which is supposed to focus on clinical research, has its funding divided between public health research, clinical research, and therapeutic trials. Finally, the lack of opportunity for collaboration. And I think much of what we've described today, the CAR T-cell therapy program work that Dr. Bellare's group has done, Professor Ravi's group has done, several leaders in biomedical research out of IIT Bombay, has happened in spite of the system. We don't have a system which enables and encourages collaboration across disciplines. And I think this is something that we really need to look at. Now, there's a lot of problems that's happened with biomedical research in India. So we can either moan about it or we can actually do something about it and come up with solutions. And I think the Cancer Grid has been an excellent example of how we have overcome some of the problems that I just mentioned. Collaboration is central to what the National Cancer Grid does. And for those of you who are not aware of this, this is a network of over 325 cancer centers, research institutions. IIT Bombay, incidentally, under Professor Devang Kakar's leadership, was actually one of the early members of the Cancer Grid. And we are hoping that this expands even further. In addition to having its main goal of having uniform standards of care across the country, one of the pivots on which the Cancer Grid was created was to be a ready platform for collaborative, multidisciplinary, multicentric cancer research. And I think this is a great opportunity for us to collaborate on that. Training is something that we've addressed within the National Cancer Grid, training individuals, physicians, paramedics, and allied professionals on how to do clinical research is something that we've done very successfully over the last 7 or 8 years. Raj was part of one of our workshops which trains researchers on how to do research, and we've had excellent outcomes coming out of that, with not just the researchers getting trained, but actual clinical, potentially practice-changing clinical trials coming out of some of the protocols that were developed in these workshops. Understanding the need to facilitate researchers doing research without the pain points like contracts, insurance, regulatory submissions, and so on, we created a clinical trials unit and a contract research organization which helps them with the nuts and bolts of doing studies. So effectively, what we are trying to create is an ecosystem which is enabling not just collaboration, but also excellent translational research which can come out and change practice anywhere in the country. I'm going to leave you with what I think is a fairly audacious idea. We have a ready network of 300 plus cancer centers and research institutions. The IITs, and I was surprised when I first started collaborating with IIT 15 or 16 years back, that IITs have probably more biomedical researchers than medical hospitals themselves. And if we can create an enabling platform where not just IIT Bombay, but IITs across the country can work with the National Cancer Grid. And I'm using cancer purely not because I'm involved in it, but as an exemplar, which can then be followed by other disease areas. We've already had requests coming in from the Diabetes Association of India to share best practices with them. This can be an excellent model to create a melting pot where we have researchers from technology, from economics, from policy, from clinical sciences, and the basic sciences to actually share ideas and come up with something which can be remarkably transformative in the way that we do research here. I'll leave you with this thought, and this is clearly will take a long time to actually implement, but I think this could be truly transformative in the way we do research. Once again, I'd like to thank the IIT leadership for the opportunity to be part of this important event, and Raj for his generosity in supporting his alma mater. Thank you very much.
Thank you, Dr. Pramesh, for your thoughtful insight. I request Professor Chaudhary, Director, IIT Bombay, to felicitate Dr. Pramesh. Welcome back, everyone. We begin the second session with our distinguished alumni, Mr. Raj Nair, BTech 1971, Chairman of Avalon Consulting, Germinate Solution, and AGR Knowledge Solution, to share his vision on the 10x GMP facility. Mr. Raj Nair's tireless efforts over 2 years to provide highly affordable medical solutions to the society have been instrumental in making the facility a reality. Can I have you, sir, on the stage, please?
I've lost the audience to tea and coffee, which I think will be, they know it's going to be better than my talk. So good afternoon, ladies and gentlemen, and all the distinguished faculty, guests here, present here today. Fortunately, I don't have a prepared speech because a friend of mine who wanted to have drinks with me there a few days ago said, don't write a speech because if you've done this out of your heart, speak also from your heart. So it's as simple as that. Okay. So I'd like to thank IIT for giving me this opportunity to use my time and money in a way which is very productive for society, hopefully very productive. And today I'm very happy to see so many of my friends and colleagues, not just from Mumbai, but from Pune, Bangalore, Chennai, etc. Some from the Middle East, just for this. Thank you so much, every one of you. From the bottom of my heart, I say thank you for participating in this happy moment. Now, somebody asked me, a lot of people have asked me in the last few days, what is this 10x GMP? And they say, we understand GMP. And I say, oh, good for you. You understand GMP. It took me 3 years. I still don't understand all of it. But good. But what is 10x? Now, normally, a good way to explain, or the best way, is to tell stories. I can't match his capability. So what I think I'll do is I'll just create a construct based on real stories and make you the principal actor in it. Okay? So let's imagine that you are a poor farmer. You are an unskilled worker in a small-scale manufacturing unit somewhere. And you're a peon in a private office. One thing that connects all of you is your lack of access to too much money, either by way of income or wealth. And life is somehow, you're pulling on. You know how in India we somehow manage. But one day disaster strikes. And one member of your family, maybe you're one of your parents, or one of your children, or your spouse, or one of your siblings, is struck by a difficult to cure disease. Maybe multi-drug resistant TB, or some other thing, or maybe one of the many cancers. And since we have people who are very familiar with cancer here, let's say it is one of the cancers. Obviously in India we will not abandon. We'll try our very best to somehow treat and save the person. So now you're in Mumbai and you've spent about 8 or 10 months in treatment and I meet you. So we start conversation and you say you spent 5 lakhs of rupees. And there's a lot of stress when you say I spent 5 lakhs. You brought your patient to Mumbai because you wanted to go to Tata Memorial where you have some of the finest doctors in terms of skill and capability. Some of the finest doctors in terms of dedication. And it's also highly subsidized. So obviously you migrated to this place or converged at this place to seek a solution for your family member. But it still costs 5 lakhs. So I ask, how did you manage the 5 lakhs? They say, oh, and you tell me, I sold my farm, or I sold my house, or I sold both. And knowing that it's going to take endless amount of time, because this is the nature of the beast. You have left your job and you come. You have no income at all to your daily life. You live on charity. And I ask, where do you live? I live on the footpath. Now, this is the story of not just one family. It's the story of more than 1 million families in this country. We have got 350 million families in this country. So 1 or 2 million is bound to be the case. Now, how can we sleep at night knowing that there are people going through this kind of ordeal day in and day out? As we speak here, this is happening. This is a reality. So I had this fond hope that if only this 5 lakhs could be brought down to 50,000, then maybe this family could have got some donations or pulled some money from here or there and not have had to give up their house or their farm or whatever it is. And when I ask you, what about the children? Oh, we've given up the house, so we've brought the children back here. They're also living with us. So education's gone. What it means is that for 3 generations, they're not going to come back to the same place. in India it is very common and we little appreciated that one major disease or accident can drive a family which is in the lower income group or which is poor into complete penury. So it behoves upon us to at least think about what can be done. Not all of us get the opportunity which IIT gave me. So I was hoping that this can be brought down to 50,000 by some kind of magic. Or I thought maybe it's possible that the treatment can be so good and so quick that instead of taking 3 years or 36 months, it can be done in 3.6 months. Now, if it is done in 3.6 months, you don't have to leave your job, bring your father or your sister or your wife here for treatment. You can take leave of absence without pay and then go back and life starts all over again, just as it was before. Or maybe if only it's possible that instead of 2 months survival because of the treatment, extension of life, or 2 years extension of life, it can be 20 years. How wonderful, this whole effort and investment in emotion and money has paid off. So I said there must be some magic solution some way, and I was surprised to discover that this is precisely the kind of work that's going on at the Biosciences Bioengineering Department and Chemical Engineering Department. I said, look, this is obviously the thing to do, do something that can help this happen. So there's a lot of research which happens, 1.5x, which means 50% better than what's going on, which is pretty good, but it doesn't move the needle. Even 3x may not move the needle. I said, how about 10x? 10 times better, that is 2 years, instead of 2 years, 20 years. Instead of 36 months, 3.6 months. Instead of 5 lakhs, 50,000. I'm just giving some numbers, okay? So, all of us understand here, if not here, that this is meaningful, this 10x. That is why 10x. Okay, and there are some amazing number of researchers. I was personally surprised that there are more than 300 PhD candidates doing research here, just in the biosciences and bioengineering department. And there are other researchers who are above that, or not above that, and there are some 40-odd brilliant young professors. Everybody is young compared to me, so even if you're 50, you're very young, okay? So there are many brilliant young professors who are driving this kind of stuff. So it was very obvious for me to say, let me just focus on this. And I happened to come across my childhood friend Ajit Javle, who's also probably 8 years junior to me at IIT, and he's an expert in this. And he volunteered his time. And between him and the team, this has happened. It took 3 years, 1 year thanks to BMC and 2 years of hard work here, and a lot of support from a department here called DRF. Nidhi has been outstanding. The kind of problem solving is unbelievable. Thank you very much. So that's 10x. I don't want to take much time, but I've got 2 minutes more. So maybe I'll tell you a story about what's the difference between a dog and a marine biologist. Anyone can guess? A marine biologist and a dog? Well, a dog wags his tail. A marine biologist tags a whale. Okay? Now, there's a lot of tagging you have to do. Tagging of people, skills, etc. to take what you have developed today. And I'm assuming that all of you are going to, many of the researchers here are going to do amazing developments as you go along. You need people to help you in funding. You need to find out what's the best way to structure your business. You need ways to figure out what business model to create, what kind of marketing you should do, or whether you should do licensing. So all these are things which are outside your sphere of expertise. Just because you have a student who is not so interested in studies, it doesn't mean he's an expert in commercial things. There's some people make such assumptions. There's a lot of things. So I said, look, maybe I should not stop at this, but just actually help to create a pool of people who are experts in all these fields and who are willing to give their time to guide people like you who are doing some phenomenal work. So that's the next phase, and I've already started working. There are a lot of people that put up their hand and said, we'll give time. Because if such amazing things are being done in a research institute for the benefit of society, we are going to put up our hands and help. So I hope that's possible. So I don't want to take too much of your time. Thank you very much for coming, and I really bless you for having come and helped. And please spread the word to industry so that industry can collaborate with us at IIT. And then magic starts. Thank you very much.
So can we have you here? Thank you, Mr. Raj Nair, for your vision and your determination to bring this facility into action. I request Professor Ravindra D. Goody, Dean ACR, to felicitate Mr. Raj Nair. I request Mr. Ajit Javle, Managing Director of Nixer Consultant Private Limited, to share a presentation on the 10x GMP facility at IIT Bombay.
Yeah, good afternoon, everybody. Thank you, Raj, for giving me and our team the opportunity to give back something to IIT. Thank you very much. So just some technical details about the facility. Regarding the objectives, some facility highlights and capabilities, and some drawings, is I will take you through this. I will not be, because we have seen the video, I will avoid the repetitions and just elaborate on the salient features which will about this facility. So if you look at the second last, the GMP facility has to be capable of carrying out as many projects as possible in parallel without the risk of cross-contamination. So this was one objective which we had, because not just one project at a time, but as many as possible in that area was one of the objectives. The facility has to meet local FDA requirements and be supportive of producing sterile batches of products for clinical trials. So we have to go to the end of the line where it can be made and used for humans. So that level of hygiene is required. Okay. The core facility has been constructed inside the existing building in the available double height space. The utilities and technical supports are located in a utility structure which is just outside the building. As told in the video, there are two suites which are the sterile suites, and there are two suites which are the preparatory suites. So at a time in each of these, independent project can be run without any risk of cross-contamination between all the four. So the two S1, S2 are the highest grade in terms of hygiene, highest grade in terms of safety for containment. The P1, P2 are one grade lower, both in hygiene as well as in containment. The other support areas like dispensing, washing, etc. complete the entire facility. The facility is accommodated and fitted in an area of around 5,000 square feet. We have a connected power of around 300 kW. We have a DG standby. We are having all the utilities. We are having compressed air of pharmaceutical grade. We are having UPS, so to ensure that all the critical instruments and the critical facilities are continue to operate without a moment of shutdown. We are having purified water, we are having ultra pure water, all what a GMP facility needs, the infrastructure is there. The facility is built using sandwich clean room partitions and walkable false ceiling. They are all fire resistant for 2 hours as per requirements of regulatory norms. All the light fittings, switches, sockets, supply, housing, return risers, utility interventions, drain outlets conform to the latest standard and specification of present CGMP practices. Is all state of art. The floor of the GMP facility is finished with polyurethane coating. Safety features like fire sprinklers, alarm systems, provision of decontamination of waste, liquid drainage, fume cupboards, biosafety cabinets of the required class are a part of the design. The facility has the flexibility to adapt to future projects with spare capacity in all the utility equipment for minor modification settings of HVAC, MEP, etc. to suit future projects which we don't know. So if you look at the plan, our video took you through this plan, so you are aware of this. On the upper floor of the mezzanine, we are having offices, storages, a conference room. If you look at the section of this facility, the lower level is the core working area. The above is the offices and the stores, etc. The middle area is the one where the utilities and the ducting, etc. is routed. The philosophy of the design is that in this S1, S2 area, all engineering maintenance should be done without entering into that area, so that you are not exposing that area to challenges of validation. So whether it is light fittings, whether it is HVAC dampers, or whether they are the blowers of the laminar air flows, etc., they are all maintained from above without getting into the core working area. This is the feature of this facility. Then outside the building, we have built one small utility building in which all the electrical panels, the air handling units, the compressors, etc. are located. So these are the key things. Anybody wants anything in detail during lunchtime or something, you can talk to me and I can clarify many things, whatever details. So I take this opportunity to thank all the team members who worked to make this facility possible. Difficult to name everybody, but I will highlight some of the people who worked at the ground level. So we are having Techno Solutions projects as our engineering consultants. They are led by Amol Sainwar, Prasad Mahamunkar, Ashish, who are here. Our MEP and clean room partitions and furniture, etc., fume hoods, biosafety cabinets, etc., are done by a company called MK Precision. And Mr. Kailash Golani, Hardik Shah, and Mumtaz Ali, they are the people who have worked for us. They are here. We have our civil contractor, Mr. Prakash Patel from Excel Construction, who has been here and working with us right through. And we have Mr. Yatin Shah, who is now going to take over the documentation portion of it. And he is going to lead the exercise into creating the protocols and the SOPs and the validation master plan. So all in all, we thank all the people who have worked, these team members who have worked for this project to create IIT Bombay's and Raj's dream into a reality. Thank you very much, everybody, and God bless. Please.
Thank you, Mr. Javle. I would like to invite Dr. G.S. Bhuvaneshwar, Adjunct Professor, Department of Engineering Science and Distinguished Alumnus, IIT Madras, former Head, Biomedical Technology, SCTI MST, to share his thoughts on the GMP facility and the significant role it will play in fostering the adoption of GMP to take ideas from lab to the market. May I have you on the stage, sir?
If I take a little more extra time, tell me, I'll stop.
Sure.
Thank you. Good afternoon, everybody. It's indeed a privilege and a pleasure to be here in IIT. I think I was here again last time in October or November, and I think January, and it's indeed a pleasure to be among my friends here. So at the outset, I'd like to thank Professor Chowdhury, Professor Ravi, and others for giving me this opportunity to be here. Because in my experience of 40 years in medical devices development and technology transfer to industry, I think it's high time IIT had something like this. Probably it's, I would say, at least 10, 15 years overdue. Because if we really want academic research to be moved through the development phase into manufacturing and sales, and I think a translation facility or a technology transfer facility is inevitable. And so I'd like to share the story of what we did at Chitra, how did we manage in the days way back, what were the challenges and what happened there in our efforts to transfer technology. Because one of the things my mentors, Dr. Valyathan, you must all heard of Professor Valyathan, in the early days he used to tell me, Bhuvaneshu, what's the use of all this research if tomorrow I can't have this product in my theater and I can't put it in a patient? And so one of the things he emphasized to many of us as young engineers was that there's no point in doing all this unless it becomes a product, which is manufactured and sold. And so in that indirect way, sort of pushing us, he made it a commitment that research is just not enough. But as engineers or as developers, we had to get the product as much, put in all efforts, whatever needed to be done to get the product into the industry and into the market. So this is just a history of, or a story about what we did at Chitra. Okay, I think the valley of death, I think all of us are familiar with, which WHO, I think, pronounced way back in 2011. They talked about the gap between the academia and the industry, and where bulk of the good research that happens, because I've seen some excellent research happening in IITs, but still very few of them really come out into the industry or into the market. So the whole question is, how do we bridge this gap? We call it technology development, technology translation, or even transfer or translation. But this is still a big challenge even today. Not only in India, it's happening all across the world. So how do we get the culture of the academic mindset to sort of interact with the way industries work, where the industry thinks and works, with a primary emphasis on risk controlling, particularly reducing the risk of their investments. So this has been the challenge. And I think the very first product that Chitra developed was a blood bag. It's a blood storage bag which is used for, when you collect donor blood and then transfuse it. It was done way back in 1984 when the product was ready in batches. My colleague was making it in batches of 100, and it went through the clinical trials. But then we were trying to find an industry, and Dr. Valyathan found a young IAS officer who wanted to become an entrepreneur. He resigned his IAS and signed our technology transfer agreement for the blood bag. And this is a classic example of what happened. Because we're making batches at 100 or 200 in the lab. And then the plant was designed to handle 2 million bags a year. That was a huge gap, and literally where a lot of problems came. And there was a time in between, I remember, I was not really part of it, but where the industry had one person who used to say, these are all jokers, plastic bucket engineers. And then the academics say, these industry fellows, they don't know anything about science and engineering. So this sort of mutual allegations were traded, but the commitment of my boss, Mr. Ramani, made a lot of difference, and eventually we managed to get it transferred. And the bags went into manufacturing and sale in, I think, 1987. So realizing this, the next time we came up with other products like hydrocephalus shunts, heart valves, and oxygenators, Dr. Valyathan said, this approach does not work. What do we do? So the main issue he found was, one is the market entry challenges that the industry faces with an Indian product. Particularly, the Indian clinicians are think that all Indian products are poor quality. That was way back. But the biggest challenges was the low duties. There are a lot of inverted duty structures those days, as well as I said, the quality of issue. So he came up with this concept of saying, he invited industry and said, we will jointly work and show that our technology works. We'll develop the laboratory proof of concept into a technology and show that it works and share the risks. And I think looking back now, this is something I didn't realize when I was actually involved in those days. But I think the important thing that he conceived, the administrative, or what you call it, the legal structure, which is I think a very critical aspect, was he started with a simple MOU instead of a technology transfer agreement. So the MOU, as you know, is a much less enforceable legal instrument. It's just more an understanding and not an agreement. And so the industry investment was kept low. That was, I think, the critical issue there. So the risk to the industry in terms of financial risks was kept low. And this was converted into a project, so that at the end of the project, if the project failed, both the institute and the industry could just write off everything without any mutual commitments. I think this is a very, very critical factor. Because one of the things we found that the industry is very reluctant to hire people because that becomes a long-term commitment. So that was one of the reasons the project mode actually made a lot of sense there. Hiring people on a project mode and keeping them on a short-term contract approach. That made a lot of difference, in my opinion. So the institute provided the infrastructure and capital, just like we're going to do with this GMP facility. And as I said, if the development fails, just a termination of the agreement, MOU, without any liabilities on either side, and then sharing of whatever was purchased resources, that would be, through some understanding, we'd just share it off and close the project. But if it is successful, then the commitment was in the MOU that industry would sign a technology transfer agreement with terms for royalty and license fee and so on, and then take it into the market. And so this has worked very well. I'll come to some of the examples. So we work with the industrial partner. This is, I think, very essential. Jointly scale up from laboratory to pilot scale. So this is the development part where you standardize all the SOPs, you standardize all your processes, you validate all your processes, you try and implement quality assurance, quality controls at steps, which does not happen in a lab. So how do we do that? Your vendor validation or supplier validations. So a lot of these issues have to be sorted out. And then we had to meet the requirements for clinical trials. That was the key issue that Dr. Valyathan wanted, is how do we make manufacturing grade or consistent quality products for clinical trials. I think this is the key, because it's unethical to put products which are not of quality in patients, products which are not well made. So that is a key underlying factor when we had to do this, is you have to do some sort of a pilot production before you can get into clinical trials. So it's sort of putting a cart before the horse, or the horse before the cart sort of issue. And then, of course, it also enabled the industry to get market seeding and feedback on the product itself. So what we did was we set up a class 3,000 square feet of a class 10,000 facility, which was what was required for medical devices. Within that, we had about, I think, 200 square feet of class 100 with laminar flow benches for the final assembly of the product. It was managed by a project team consisting of both the R&D side as well as the project team. One or two could be from the industry also. And then we produced quality devices. We supported the clinical investigation, not only with the devices, but sometimes even with the data acquisition this is. And then we developed all the technology transfer documentation, all the process SOPs, the procedures, as well as the policies. They were all developed during this time. And then we trained the industry core people, or the project team were then hired by the industry when the technology transfer happened. So the overall, the benefits to industry was they could observe that the technology really worked. We had a scaled up process. The technology really worked. They could be satisfied with the clinical performance and safety of the device, which was very, very critical. This was the whole concept of the technology proving. Carry out market seeding and determine product acceptance, and then have all their own pricing strategy. I think one of the things that most academics, we all forget, is that pricing a product and putting it in the market is a strategy. It is just not saying X over the manufactured cost or something. It's always a strategy. And it all depends on the brand value, it all depends on various factors, depends on the competition and so many things. So it sort of helps the industry to have all their pricing strategy when they enter the market. And of course, they got a core group of fully trained staff, and then with complete quality management systems and documentation in place. So some of the, as I mentioned, the unique framework, which is very critical now looking back, was the MOU instead of a formal technology transfer agreement. It was more informal and managed in a project mode by a joint team. There was no formal employment by the industry. I think this is one of the fears they always had. They didn't want to employ additional people for this project. So there was no formal employment by the industry. And fixed upfront limited commitment by the industry. I think these were some of the factors which helped us get the industry. But even then, I think in a couple of cases, I had to run behind the industry to get them to come and work with us, and then offer them SOPs and so on. But if you really want to transfer, you have to run after the industry sometimes. For the benefits to the institute, of course, we could support the clinical trials with enough clinical quality products. That was very, very critical. It reduced the time from lab to market. A core group of staff got fully trained, so I all learned about scaling up, standardization, process validation, all that, though I was part of the academic side and doing laboratory development. And then, of course, we could sort of transfer the know-whys, many of the underlying science and engineering principles which are there in the product. This also we could help transfer it to the industry. And then it also in future process and product improvements, we much more easily carried out when you know what you're doing and why you're doing it. So some of the steps was evaluation of alternate sources of raw material. I'm sorry. Where have I gone? Yeah, alternate sources of raw material. Then development of process equipment. Sometimes we had to do that. And development, training of manpower, standardization of processes and quality management systems. And sometimes you have to do some work and motion study, basically to optimize your processes. And then the development of the quality assurance procedure, which is a major requirement when you really want to get something into manufacturing. This is some of the successful projects we ran. The first was a bubble oxygenator and a cardiopulmonary reservoir, which was part of the heart-lung machine during open heart surgery, which was then transferred to SPIC in Madras. They set up a subsidiary company eventually. The heart valve, where I was primarily directly involved with TTK Pharma. Then the hydrocephalus shunts, another product I was involved in the development. Concentric needle electrode, membrane oxygenators, wound dressings, and so on. So we had about 16 products which went successfully into the market. So do I have time? For 5 minutes? So I thought so that's the sort of process. Of course, there's a lot more in it to discuss, but briefly, I think the framework, the administrative framework, or how you negotiate with the industry, I think that framework is very, very critical. Looking back, I think it's something I didn't know when I was really working hands-on, but now looking back on it, I think that was a very critical aspect of the whole concept. So I would stress that if IIT Bombay really wants to, how do you commercialize this? I think the framework of how you engage with industry, I think is a very critical factor to bring in success. And many times this framework has to be fine-tuned to the product and what is involved in the project in the development scale. So when I was head of the research wing and when I was dealing with industry, we used to have sort of customized agreements where I say, this is the responsibility of what institute side we will do as part of this project. These are the responsibilities of the industry side, and these are aspects where work elements which will be done jointly together. And unless that is clearly identified in the MOU, invariably there's a lot of confusion between the industry side and the academic side. So detailing down particularly the work elements and work packages as a project, I think, is equally critical as part of the overall framework. So some anecdotal examples of the challenges we faced, I think many of them will come up again. I think the first was a blood bag, no, though not really part of the technology scale-up, was really related to labeling, not really the product in itself, but the labeling became a big challenge. One of the problems that came early on was, we had these blood bags in batches being made, and you can't stick paper and then the bags were steam sterilized, autoclaved. And when you stick the label and then autoclave, and then when it comes out, the paper is really all sort of wrinkled up. And this primarily because the coefficient of thermal expansion of paper is very low compared to the coefficient thermal expansion of PVC, which is about 10 times or even about 15 times that of paper. It's a very simple physical problem, but it looks very ugly, this method. So we had to stick the paper after the sterilization. Though it's outside, that was considered okay. But the problem is many of the papers have fungal spores still present. And since you have CPD, the solution inside the bag, which slowly the water diffuses out, after about 3 months, some of the bags were returned from the blood bank, saying there's a lot of black things behind. So it's not really inside the bag, but it was the fungus was growing behind the label, between the label and the PVC, and it was looking very bad. Obviously, nobody would use it. And how do we solve it? This became a big issue. And after some research, hearing things, we found that I think the Fenwal bags, not the Tuta bags, which are made in US, they had solved the problem by sourcing the paper from a tree made in Canada, which didn't have any fungal spores. The question is, India, what do we do? Where do we go and find a paper which doesn't have fungal spores? This was a big issue. And then I think one of my seniors, he came up with the concept of pasteurization. So we used to pasteurize the bags after the label is stuck at about 70, 75 degrees, and then put them in a second bag. And later, I think the other thing was tried was to gamma ray sterilize the paper, label itself separately before we are sticking it on. And then plastic label still continues to be a problem world over. It's not simple, it's not very easy as it is, but most of the problems have been solved. The other problem was in distilled water. We're using a lot of distilled water for cleaning our components before assembly. And we were using a 20-liter still, actually, primarily. And that's the condenser. And this was during our hydrocephalus shunts. I think we started sometime in September. The project we had made by about February, March. The next summer was starting. We had made about 5 or 6 batches, and all the batches, every batch we sent 3 samples to the microbiology lab for sterility testing, and everything was going okay. They were all released for clinical trials. Then suddenly the 5th or 6th batch came back unsterile. Now we started scratching, how did this happen? Because we were rigorously following the same steps. It was March, it was very hot in Trivandrum. And our GMP facility main area is not air conditioned. And so we did a lot of testing, so we had to take a lot of samples and do culturing. And finally, the culprit was found to be the distilled water we're using for cleaning. And we again changed the tubes, we cleaned the distill, we did some samples, again came out unsterile. So a lot of work was done from microbiologist side taking samples. And then we found that we had a nidus of infection sitting inside the condenser. Now, this is very unusual. I mean, you're talking about a condenser where the water temperature is around 70, 80 degrees centigrade, but still we had a nidus core of bacteria sitting there. So we had to do a lot of steam flushing and all that too, because it's not very easy to clean that condenser. But this was a very challenge. It took us at least about, 2 months we spent in trying to solve this problem. And I'm sure these challenges will come up whenever you're scaling up and you're trying to develop a manufacturing process. Another very simple, but I think Jayesh should appreciate this better, is the HIP sol-gel process. My colleague Verma had developed this in a bioceramics lab. It's a bioglass composite for anterior use in dental practice, basically for bone filling. And we transferred it to an entrepreneur. It was IIT Delhi alumnus who took the technology. And he set up his factory near about 40 kilometers north of Chandigarh, somewhere in the foothills of Himalayas. But that's critical. I think it was at an elevation of 2,000 or 3,000 feet. And then we transferred it. He sent his people. We trained them. 3 batches were done. We usually help them by testing the batches, making sure everything's fine. They've understood the processes. Then our technician also went to the factory and made sure everything's worked. 2 batches they made. Again, somewhere in July, August, everything was working fine. Then he came back. And then suddenly in November, we got a call saying, sir, the sol-gel, it's not precipitating. It was, I think, early November, early December. Now, we're all sitting in Trivandrum scratching our head why it is not precipitating. And then, so finally we had to send our technician. I think he went there end of December or January. And then we found that the room temperature, which is what the specification for the process, the room temperature was 16 degrees compared to 24 degrees of Trivandrum. Just 1 minute. And a simple thing, so one of the things in GMP or in all quality management systems is you can't have vague numbers, like room temperature for ambient conditions. So finally, we had to change the documentation. So each time after that, we say, when you say room temperature, is it 22, 24, 26, 28? What's it you're talking about? And what is your range? So these are classic examples that happens in this one. And the last thing, just a couple of minutes, was related to my heart valve. We transferred it to TTK, everything was fine. Of course, the metal cage, which is the core component, the most expensive component, which is machined from a cobalt alloy, a very hard alloy. It's an expensive material. So that was the most expensive component. Everything was fine. The TTK were manufacturing it. Suddenly, one batch, we had a general machine tolerance of plus minus 50 microns for most of the major dimensions of the valve component. And so suddenly, this TTK person, the project leader, he came to me and said, sir, I have 5 cages which are just 10 microns above the tolerance. It's being inexpensive, difficult to get machining in those days. I'm talking about the mid-90s now. And CNC machining was still not easily available in India those days. So do you want me to reject? It's going to cost us a lot. So I said, because the valve height, which is about 5.2 millimeters, and our tolerance was plus minus 50 microns. Now, if it goes 10 or 15 microns more, it's not going to make a big difference to the valve functioning or anything, but do we accept it or do we reject it? And this was a big question. How do we solve this problem? So I agreed it was a problem because it added to the cost of the project or the cost of the products. And so then my colleague went a lot of research into the thing and found this concept of this defect management. I think there are policy strategies you can involve in how you manage defects. You can classify your defects as this is what we found as A, B, C, from low risk to high risk. And you can then have a policy saying this and this defect you can have and accept it as acceptable, but this particular defect, if it is there, okay, it's rejected. So we dropped this table of defect management policy and then we sorted out the problem. So these are critical issues where as academics, most of us don't understand that how do we solve these problems in manufacturing, because they all add to the cost, they can add to the rejection issues, or how do you manage rejects also. So see that these are some of the challenges that many of you who are partly academics who are going to be involved in the fabrication, you're going to see similar problems or many of these problems. And I hope, I'm sure that all these challenges can be overcome provided we understand the problem and how do we solve it. So thank you very much for your patience.
Thank you, Dr. Bhuvneshwar, for sharing your thoughts with us. I request Professor Goody to felicitate Dr. Bhuvneshwar. Sir. Now we begin with a series of presentation on the different types of products that will be manufactured at the 10X GMP facility by our faculty members of the Biosciences and Bioengineering Department, Professor Bilare, Professor Tayalia, Professor Sen, and Professor Srivastava. Can we have Professor Bilare on stage?
Thank you. So I speak from the perspective of a professor here, and the greatest joys of being a professor here, of course, is the great students you have and the facilities and the papers we publish. So what drives us really is the innovative aspect, the academic freedom that it gives us to pursue various things. But lately, what brings us the biggest joy is the fact that our research, which typically is in papers and occasionally in patents, sees application, right? So that's actually the biggest joy of having an academic environment, which now is going to be further enabled by this facility. So I'll spend just 2 minutes to tell you about the kind of products that we've worked on that has been enabled by virtue of having GMP facilities here. So we all know that broken bones heal quickly, except when there are issues. One of the issues is if the break is long, it's of a large size and therefore difficult to heal. It's like a bridge which is too long to build and needs special support, and often bone grafting is done. So one aspect of the work that we've done as a R&D project over the last 10 years or so, with multiple students contributing to it, is to build scaffolds of the sort shown in the left of this view. These are spongy materials which which facilitate bone to grow very quickly. So this is a process that we've developed. The material is new, patented, published, tested widely. But what it really needed was, in order to make it go first in human, which is a key step in any transfer of technology of a biomedical device, is a first in human trial, and that really required a GMP facility. And we had a pilot project to build a small GMP facility which enabled this, and thanks to this, we were now able to expand this into a first in human clinical trial, a pilot trial, which then led to a startup which took this up, then a startup led by the student who was working on it. So it's great to see our students, PhD students especially, and this one was led by Dr. Kunal. And we now have a dozen employees of this startup, all of them highly qualified, who are now contributing towards this effort, a multi-center trial which is used in the application which I show on the left side of this slide. And we are now working on newer products which will enable larger bone grafts to be done. This includes technologies such as bioprinting and biodegradable materials. And this is a startup now nucleated at SINE and doing a multi-center trial at 10 top-line medical institutions across India, over 150 patients. And kudos to this team which is now, of course, independent from us, but has taken this technology and moved along thanks to that little GMP facilities. We need to multiply that 10 times, and that is what now we are setting out to do today. Another product that's in the pipeline is a bone screw, a resorbable bone screw that some of our students work with. Professor Rohit Srivastava's group was involved with this as well. And this is another product that is now being manufactured, and this will be one of the early products that will be made in this facility, in this new facility, which we have inaugurated today. Yet another project which one of my PhD students, now postdocs, is working on is this problem of addressing pelvic organ prolapse and urinary incontinence, which plagues older women, some older women. Yeah. And this requires a product, a biodegradable product, which can be used surgically to help support these organs. And this is another startup which has come out, which is eagerly waiting for this facility to get rolling, because we are ready to take trials. Even some animal trials now require us to do the manufacturing in a GMP, but certainly the human clinical trials. So thank you for your attention, and we look forward to many, many more such products. We have a very rich pipeline of R&D which is there and which can exploit this facility. Thank you.
Good morning, everyone. First of all, I would like to thank Mr. Raj Nair, Mr. Ajit Javle, and the entire team of the ACR office who has helped, bringing this GMP facility actually into completion. And, we look forward to actually using it for some of our products now. So just to give a brief background, I myself am from a material science background. I'm an alum of IIT Bombay, same as Mr. Nair. And because of that background, and now that I'm part of the bioscience and bioengineering department, it has allowed me to work on some of the technologies which allow interdisciplinary research, and I'm going to talk about a couple of them today in my presentation. So the two things that we are really looking forward to conduct in this GMP facility is, one is the dermal implant, which is related to the wound healing project, and the other, so this is how the implant looks like, and how, and the key aspect of it is that there is no size limitation for this implant, and this is sort of rolled on an arm, and we can make it bigger than this as well. The other product that I'm going to talk about is this for cancer immunotherapy, and here is, it's a cryogel-based product, and here is just an example of how a tumor can be reduced to smaller sizes. This is specifically for a solid tumor, and I'll talk about that in a little bit as well. So moving over to, just to tell you broadly about what my lab does is, as I mentioned, we work on biomaterial-based technologies, and we develop, based on different composition and architecture, we make different kinds of 3D scaffolds, and we use them for skin tissue engineering, one which related to the dermal implant project, and also the immunotherapy that which I'm going to talk about now. So these are the two application-oriented projects that we work on. The third one is where we actually use these scaffolds also to develop some in vitro systems, and which can be used subsequently for drug screening or even understanding the material, cell-material interaction. So the first one, which is the bilayered multifunctional wound dressing that that we are interested in using this facility for. So here's just like a brief overview of what these dressings are going to be used for. A couple of applications is the burn wounds and the diabetic wounds. And as you can see here, there are different stages of burn wounds and diabetic wounds that are there. And specifically for diabetic foot ulcers or burn injuries, we need a product that can cover a large area. The current solutions that are there in the market are largely cotton gauzes and such gauzes, and most of them, they have only an antibacterial property, and they do not have the moist environment which is required for good wound healing. And typically, they also have a non-biodegradable layer on the top. So what our strategy is to develop a bilayered, multifunctional wound dressing, which will have some of these properties which will allow a better wound healing. And apart from that, it will also help in the absorption of the wound exudate and maintain the moisture, which will help in a better wound healing capability. These are some of the major players that are already there, and because of all of its properties, in addition to that, the way we are making it is also going to be affordable. So here is just a little video of what this wound, the dermal patch looks like. It has this top and bottom layer. The top layer actually prevents the microorganisms from invading, and the bottom layer actually helps in the absorption of wound exudate. It has some wound healing agents which are absorbed in the bottom, which allows the proper wound healing of the system. And these are some of the conventional wound dressings, based on cotton, which make it very bulky, and they require, frequent changes, which makes it difficult for the patients to do it regularly, and it also makes it not very cost effective, as they have to do it very regularly. So these are the dressings that we have developed in our lab at IITB, and they allow easy peel off, and they also allow the absorption of wound exudate without making it very bulky, and they provide an adequate moist environment for the wound healing. So so the other thing that I want to talk about is the material-based CAR T cell therapy that we are, that we are trying to develop in our lab. So so we've heard a lot since this morning from various speakers about the CAR T cell therapy, which has come into market recently. So for those who do not know the principle, on what it works, so in a CAR T cell therapy, the T cells are taken out from a patient. They are modified ex vivo. They are modified in the lab using certain vectors, and they have to be grown in millions so that they can be reinfused into the patient's body so that they can recognize the tumor. So currently the CAR T cell therapy is mainly for hematological malignancies, and here is basically a broad statistical data on these T cell therapies, how many of them are currently in the clinical trials and market for the hematological malignancies. There are very few for solid tumors. It has not been very successful for solid tumors so far, and some of the other, such as autoimmune and other diseases, these are the cell therapies. So the main drawback of CAR T cell therapy, although it has been very successful for the hematological malignancies, is that it is not yet available for solid tumors, and it has a very high cost. And of course, it's 3.5 to 4 crores in the US. It has been brought down now that it is commercially available to less than 50 lakhs in India, but still it is a high cost. Further, the way, as I mentioned in this system, how it is isolated and it has to be expanded, it takes 2 to 3 weeks for it to reach to the patient. So it is quite time consuming, and because of making it for personalized, it involves all this manufacturing and transportation cost to it. So our strategy here is to actually use, again, our biomaterial-based strategy, implant this scaffold, which is based, the vectors that actually are used to modify the cells ex vivo. We plan to implant this scaffold inside the body of the patient near the tumor, so that the cells can actually come to the scaffold, they get modified, and they can actually recognize the tumor in vivo itself. So what this saves us, that one, it helps with providing a localized therapy for the solid tumors, and the in vivo modification makes it a shorter process. So we don't have to go through this 3 weeks or 4 weeks of long wait to reinfuse the tumor cells into the patient's body. And because of this, it again lowers the cost of the therapy. So that is the hope that we want to pursue this particular technology and see where it's going. Again, here is the data. So for both the projects, we have conducted the preclinical work in animal studies, and now we hope to use this GMP facility to basically produce some data that will allow us to take into further, to take it into commercialization and hopefully bridge the valley of death that we talked about. So this is just a list of patents and publications related to some of this work, and that's about it. Thank you.
Yeah. So I'm a mechanical engineer working in the Department of Biosciences and Bioengineering, and the perspective that I take is to look at how cells physically interact with their surrounding tissues and how this regulates the processes. So two of the projects I'll be speaking about are look at the critical interactions between cells and their surroundings and how disease conditions perturb the properties of the cells and continue in disease progression. So diabetic foot ulcers is of course a very debilitating condition, but it is a very multifactorial disease, and current practices beyond just trying to manage the disease, there is no active therapy. So we had actually jumped into trying to get a molecular level understanding as to what is the reason why diabetic wounds don't heal. And through a collaboration with a clinician at Nair Hospital and getting hold of fibroblasts from discarded skin tissues of healthy and diabetic patients, we identified that one of the potential sources is the presence of a very thick coat of glycoproteins and glycolipids, which are commonly referred as the glycocalyx, right? And what we found that if we disrupt this glycocalyx, not only are we able to prevent the death of the fibroblasts, which make up the dermis, that is a critical component of the skin tissue, but it can also promote their proliferation and deposition of tissue, thereby regenerating the dermal tissue. And so this is a bioactive element. We are now incorporating it into tissue adhesives. So we plan to develop bioabsorbable tissue adhesives with one of these enzymes as the bioactive element. We have confirmed it, validated its efficacy in patient samples, but not in animal models. That is the stage where we are at. We have also secured a grant from ICMR for a collaborative project with AIMS doctors, where they are going to manufacture these strips and test it on patients to see what is the safety and efficacy of this. Now, interestingly, in the second project also, glycocalyx turns out to be one of the therapeutic targets. So in cancer cells, studies have shown that they have a bulky coat of this glycocalyx. Now, this glycocalyx acts as a physicochemical barrier, which really restricts the uptake of drug. And among the many proteins, so mucin, which is a prominent glycoprotein, is highly overexpressed in multiple cancers. It can be mucin 1 or mucin 16 or other mucins, as the case will be. And the strategy that we are following is, we have seen that in experimental systems, if you treat with drug, then you have an increase in the proportion of cells which are mucin high, and they also tend to be drug resistant. So the approach that we are taking is to basically develop a nano formulation which incorporates mucin 1 guide RNA, the idea being that after initial disruption of the glycocalyx, we are actually going to knock down or knock out the expression of the mucin, thereby enable increased uptake of drug. Therefore, you can have better therapeutic efficacy with relatively lower doses. So this project is, of course, at a relatively earlier stage, but we are keen on taking forward the first project at this point of time. Thank you.
Yeah, so thank you very much for the opportunity. Thank you, Rajji, for bringing this facility to our department. It started out when I was head of the department before Professor Balaji, and Professor Suhash Joshi had brought Rajji to our department. So once again, thank you very much, Rajji, and Ajit ji for helping build this facility along with Rajji, the Dean ACR team. I'm going to talk about two products which are going to move into the GMP facility, and I'll give just a brief overview. So we've set up 5 startups in the last 1 year. 3 of them are already funded now. One of them, the one that I'm showing here, we are looking for funds. Of course, we'll get it. We are confident about it because we have a diverse portfolio of products in the startup. What was crucial to us was this GMP facility, because we have now completed all our preclinical studies under GLP conditions, which means that all our material is now ready to move in GMP, which will now make it move ahead into clinical trials. We already have quotations from 2 CROs to take these products ahead into clinical trials. What is crucial for us to understand is that the GMP has been built in such a way that it can actually do simultaneous projects at the same time. So this is what you saw from a few of our colleagues, including Professor Bellare, and we hope that we'll be using the GMP to its fullest capacity. I set up a startup last year with one of my postdocs in the lab. This is called MedInotech, and the portfolio is pain and infection management. So both these products we are planning to move forward into clinical trials this year, and we have already started talking to potential investors to put money into the startup so that the clinical trials can happen. If you see this product, the first one is infection management sponge, and this will have to go through a rigorous testing process in humans before it can be made available for clinical use. The important part about this is that the idea came from an orthopedic surgeon, and this is why there is potential interest in the orthopedic surgeon community for using such a product, which can lead to having, having lesser infection post orthopedic surgeries. The second product is pain management patch. This is a unique product. Across the world, there is none like this. We have seen many of these passive delivery patches, and there are options of using an active delivery when you use some active mechanism like sonophoresis or ultrasound-based delivery for or even electric current to actually move drug across the skin. This is somewhere in between, that it will also have a patch like you see with passive delivery patches. Think of a nicotine patch, but having small perforations that can penetrate the skin, the upper layer of the skin, but then that can have 100% of drug delivery compared to a nicotine patch which will lose 70% of the drug over the period of time. So we've actually made these patches, we've tested them out in animals, and we are starting to see very, very good results with the patches. So we are hoping to make a full portfolio of these products in the company, which is going to look at pain management. And these are going to use microneedles. The microneedles, we already have a process to scale up, and thanks to this facility now we have all the equipment to scale up the microneedles in the GMP facility, which will now make it available for clinical trial use. That's all I had to say. I don't want to take too much time. Thank you very much.
Thank you, faculty members, for your in-depth presentations. Next, we move on to the panel discussion on the theme of world-class medtech manufacturing through GMP and QMS. For this discussion, I would like to call Dr. Pawar, Dr. Pramesh, Dr. Ganga Khedkar, Dr. Bhuvneshwar on stage. And to moderate this session, I would like to invite Professor Bellare, Chair Professor, Chemical Engineering, IIT Bombay.
Comes up.
Hello.
I want to just share a small trivia, but it's not very trivial. Can you guess what is the current net worth, average wealth of a biosciences, biotech professor in the US who's gone beyond GMP? The average wealth of a biosciences professor in USA who has gone beyond GMP into manufacturing. Any idea? Take a guess. $100 million is the average net worth of a biosciences professor. But let that not be the motivation for your research. Please do some deadly stuff.
Thank you, Raj, for setting our expectations right. Yeah, we'll have to keep a slice in it for many of us. Yeah, thank you. But let's, I know we are all acutely aware, cognizant of the time overrun, but let's have some directed discussion now, and hopefully we'll have some time at the end to take open questions. So I'll request, few minutes of open mic for each of our panelists, keeping in mind that the quality systems and the GMP manufacturing has been a cause for concern for many in the pharma sector in particular, and we've received, bad press on this, and in fact, factory shutdowns and so on. Of course, this is at the industry level. So that's one thing which the quality issues directly impacting production. And associated with that is the fact that in terms of biomedical devices and medical consumables, we have been laggards as a country, whereas you can see there's some very innovative work going on here. So keeping these two factors in mind, I'd love to have your opinion, sir. And let me just go in order here, because Dr. Bhuvneshwar has been a leader of the one institution in India which has actually bucked this trend and actually brought out products. So please go ahead.
Thank you, Jayesh. I think let me first say what is quality. I think that's the critical issue. Quality is obviously the product's parameter which ensures its performance. When you say GMP, GMP is essentially a terminology which I think mentioned, which is primarily used by the pharma industry and the biologicals, the vaccines and others. And when it comes to medical devices per se, we talk about ISO 13485, which is the international standard for quality management systems. Now, GMP is also in a sense a quality management system in its own way for pharma. But when it comes to the eventual quality of a product, the quality management systems are sort of the foundation, the basic requirements, which ensures reproducibility and repeatability of the product's performance. So that's one aspect. But I think we also need to look at the initial design, the science behind it, the engineering foundations, the standardization of the process, which is all part of GMP. But essentially also including the quality assurance that goes in, as well as vendor management. So there are multiple parameters involved. So GMP is, I would say, the very basic requirement. The facility has to be clean. The facility, the cleanliness, the air quality, and the handling processes, how the materials move inside the facility. I think these are critical. They have been partially designed into the facility, but the training of the manpower who do the job is equally very, very important. I'll give you an example of the sort of mindset in India we started. This happened in our blood bag factory, which was a GMP clean room where everybody had to change into clean dress, as a minimum to go into. But the factory boss, the plant manager, thought it doesn't apply to him. So in fact, in the early days as black, he refused to change and go into the factory until I think the DCGI people came and put a bit of a fright into him, saying, we're going to reject all your blood bags. So there's a mindset issue there amongst all. Particularly the challenges in the clean room manufacturing is when people have to come out for their coffee and tea and lunch and breaks. So when you come back, do you again change and go back, or do you take the tea and coffee inside? Do you allow that? So these are some of the other issues that do come up. So these are the thoughts that come into my mind. It's sort of a multi-parameter complex issue of maintaining quality. But I think you have to develop the practices and the culture for that. Thank you.
Yeah, thank you. So I guess we are addressing that. We have Mr. Yatin here, who's going to be helping us along with all these SOPs and the kind of hopefully leading towards a 13485 sort of a QMS system. But let's have your views, Dr. Pawar.
Yeah. First of all, thank you very much for inviting me here as a part of a panel here. So as far as GMP is concerned, I would put it as 5 Ps. That is, first is people, that is staff who will be working. They are very important. So that comes under human resource. They should be properly trained, competent to handle the situation. That's most important component. Another is product. I mean, there has to be clear specifications to handle the product at all phases of testing. Another is process. I mean, there has to be a fully documented process. I mean, as sir mentioned, there has to be SOPs for each and everything, starting from entry to exit to actually handle the product. So all those process requirements are very important. procedures for everything, SOPs as it was mentioned. There will have to be SOPs for each and every step, and they need to be followed. They need to be reviewed annually, so there has to be a review process, amendment of SOP as the process progresses, and the last premises, that is infrastructure requirements. I mean, there needs to be requirements which need to be fulfilled as per the GMP guidelines, which includes environmental conditions, in engineering aspects, as far as equipment are concerned, all the calibrations, their maintenance and everything. So all these aspects are actually important parts as far as GMP is concerned. Thank you.
Thank you. Dr. Pramesh.
Thank you.
It may not be on. Just reiterate mic.
Thank you. I think I briefly spoke about this when I spoke earlier this morning, that we are far too content being 90% good, and the last 10%, which is often admittedly a very difficult bridge to reach, is the area that we often cut corners on. And while I have no doubts that this particular GMP facility will not do that, what we need to ensure is, as consumers, and I'm putting myself in the place of the consumer, is to expect the same level of high compliance to quality even from the industry. Because bad press is one aspect of whatever we heard about the eye drops and the cough syrups, but let's finally look at the end user of the product, who's actually children and people dying or losing their sight because of that. And I think unless we as consumers demand this of the industry, we are failing in our duty as citizens of India. And often I find this a little lacking in the industry as well, when they expect the fact that because we are Make in India, there can be some compromise on the quality. And I think this is an extremely dangerous path to take. And I think it behoves all of us to ensure that we demand the same high quality, because here we are dealing with patients' lives and various other functions that they have. So.
Yeah, absolutely. I think that's a very important point you mentioned, Dr. Pramesh. And this is something that we will have to imbibe as an academic institution, where often, attitude of jugaad is celebrated. So there are, of course, instances where it should be, but in terms of quality, I think we have to ensure that the SOP is adhered to, and we look forward, sir, to your guidance and your policing to ensure that we do that. I think this is very important. It's better that we do this internally rather than have DCGI, CDSU or the FDA pull us up here. So this is a cautionary note, so thank you.
And Jayesh, before you move on, I think this is also an opportunity for the GMP facility here to ensure that the SOPs that are followed here are also transferred to the industry partners, because you're not just transferring the technology, you're also transferring the culture of quality even to the industry partners.
Absolutely, sir. That's a great thought, sir. And I think this should be part of the technology transfer sort of a packet dossier that we should build whenever a product enters. So we should keep all this important point in mind, that culture should get not only the culture but the knowledge and the SOP, and this can be the initiation of a 13485 process at the potential industry. And it's of course great to see that so many of our own students and staff members here in some sense are the entrepreneurs who will take this ahead. So hopefully it will be not only the documents that get transferred, but the know-how and the brain how also that goes ahead. Thank you, sir, for that thought. Dr. Raman Gangakhedkar, sir.
Few things. No? I agree with sir when he said, there are one-time activities which you do, your building, your planning that went around, that part is fine. But when it comes to the rigor of maintenance, then the issues start. I think it's a mindset problem. No, more often than not, you find that people tend to take pride that I did this, but still didn't have any problem. No? That's something which must be avoided. That's number one. In India, there is another problem. No, whether it is clinical trials, whether it is pharma industry, you will find documentation is one challenge that we cannot put to so much of rigor that will perhaps ensure that US FDA, when it comes, most of the things that tend to come in 483s, that is related to documentation errors. Now when it comes to, because I have graduated to storytelling part since morning, how important it is. It is extremely important. And as I said, I do not want to give specifics, but there was a time when there was a big scare of a virus getting introduced just because of mistake at the industry level, and the entire country was threatened at that time. Though we could manage to handle the whole stuff well, even punish the manufacturer, which is not so easy, because that news can leak so easily and the whole country would have had been under fire, which Shailesh is also aware of. No, but I don't want to talk about it. It is only to emphasize one point. Shortcuts for providing a medicinal intervention can be extremely hazardous. We must refrain from it.
Thank you, sir. So let me go around once more before we open out. And this is, to question why more medical products are not, innovative medical products are not coming out into industry in our context. Yeah. So yours has been an exception, sir. So maybe from that viewpoint you can address that. I mean, you told us some reasons for your success. I think that MOU idea rather than a technology transfer is a great way to begin. But what should we do and what should others do so that we don't fall in this valley of death?
I think the primary issue, as I mentioned, the root cause is the lack of investment strength, deep pockets within the Indian industry and medical device industry per se. I'm not talking about the pharma, but the medical device industry is still extremely small in India. So it's a question of whether we have the critical mass for it to grow. Secondly, the government policies have not been really encouraging medical device industry to go. Even today, in spite of medical device industry being one of the 8 in the Make in India program, it has not got the sort of attention that, say, defense or cell phone technologies or others have got in terms of governmental policies changes.
Yeah, PLI and so on. Yeah.
Not just the PLI, but it's also the tax structure. So the tax structures on medical devices don't really encourage medical device. Second aspect, which really again impinges why industry is not coming forward to take technology, is the regulatory system. We have a poorly developed structure. We have been talking about it. I've been involved in this discussions right from 2003 when ICMR convened the committee.
You drafted some of them, right?
Yeah, I drafted some of them. But still, we are still talking about an effective medical device regulatory system in India. I'm sorry to say it openly, the health ministry continues to play a dog in the manger policy. Neither will they bring in an effective system, nor will they allow it to become independent and set up as independent regulatory authority. And unfortunately, the government, I would say maybe even the PMO's office or whoever it is involved, has not really moved into this effectively. That's the other aspect. Because regulation is a very critical aspect of ensuring, when we talk about quality of products, who says a product is really safe? Who says a product is acceptable for the clinicians to use it tomorrow? So unless we have an effective regulatory system, which we can rely on and for the public to rely on and say that, okay, this is approved, so it's safe for me to use it or safe for me to have it used. That's the underlying thing which is missing in India today. We tend to rely on CE mark products or US FDA's approvals eventually, and that's not helping the industry in any way.
Thank you, sir, for that viewpoint. Shailesh?
Yeah. Thank you very much. As far as my views are concerned, in India, applied research in medical field needs to be fostered. As far as I do work as a auditor on the behalf of WHO as well as NABL, I visit several laboratories in our country. So as far as laboratory infrastructure is concerned, when we talk of developing something, then at least we work in the lab, we develop something, but those laboratories, their documentation and everything is probably not as per the guidelines. and then we do something and then it doesn't end up in product. It doesn't go as far as industrial collaborations are concerned. So there are many things which need to be improved as far as laboratory infrastructure is concerned in the area of medical research. Further, I would also like to mention that whenever there is, as far as academia and industrial research is concerned or collaborations are concerned, there is a lot of scope for improvement on that front. So I think these are some of the areas which I would like to highlight. Those need improvement.
Great, thank you, sir. So, Dr. Pramod, you and your institution has been really in the forefront of innovations at a clinical level in many treatment practices and so on. So, can that now move up and down the treatment ladder to these devices and consumables and the kind of drug therapies that are needed? What is really needed for us to be innovative?
So I think one of the filters that we run through, whether it be the Tata Memorial or the National Cancer Grid, is what is the likelihood that this will actually benefit the end user at the end. And that is why we consciously took a decision when we are funding studies or research through the Cancer Grid, it needs to pass through several filters, one of which is that it needs to be something which is common or unique to the country. The second, which is that it has to be multi-centric, multidisciplinary. And this includes even centers like the Tata Memorial. We don't want single center studies anymore because one, they are inefficient and take much longer to run. And second, the results are not really generalizable. If you've done your entire study out of Tata Memorial, your results are applicable only to the kind of setting that Tata Memorial is. Unless you get settings of various levels of expertise, you're not really going to make that generalizable. We have a very strong mandate to share data in the public domain. So anyone who gets funded through the Cancer Grid has to share their raw data in the public domain so that other researchers can mine it and come up with newer findings. But having said that, coming back to your original question about why aren't we translating better, I think it comes down to funding and the lack of interdisciplinary collaborations. We might cry ourselves hoarse. The very fact that we are here today in 2024, thanks to one individual who decided to fund the first GMP facility in an academic institution in India, speaks for itself. We are a country of 1.4 billion people, thousands, if not tens of thousands of research institutions and healthcare providers, and we have the first academic GMP facility starting today. I think there is no other example of how we've allowed the lack of funding to restrict the kind of research that we are doing. The second point is interdisciplinary collaboration. We live in the same city. We are probably an hour apart, and in today's morning's traffic, it took me 40 minutes to come from Tata to here. I don't think we've even scratched the surface as far as collaboration between these two committed organizations working on biomedical researches. So if that's the case with institutions like IIT and Tata Memorial, just imagine the long path that we can take, how much more we could do if at all, if only we could get into this interdisciplinary collaboration. And I think fertilizing minds through different ways of looking, just looking at these 7 or 8 projects that I heard today, opened up my thoughts to so many ideas that we could work together on. And I think getting that melting pot where we get all the stakeholders, getting involvement of technologists, engineers, medical doctors, paramedics, policy makers, bureaucrats, economists, is probably critical in my mind.
So just to follow up on that, because your National Cancer Grid has been a sort of an exemplar in doing things right here. So is there two follow-ups on that? One is, can we take some of your best practices to examine the kind of projects that we would like to pass through this system? Because I'm sure we'll have several of those. That's one. And second is there some possibility of the kind of collaboration you just talked about by having some kind of a grid network of the sort is that something that that we could examine together?
The short answer to both of these questions is yes. I think we'd be very, very happy to share these best practices. I'm not by any means considering that these are the perfect solutions, but a lot of thought has gone into these filters, and we're very, very happy to share these best practices with anyone who's interested in this. And I think what I mentioned earlier in my talk today as well, if we could use this opportunity to actually create this hackathon, if you want to call it, get all the people together, throw around 25 ideas, even if 2 of those 25 work, I think we've done a great service.
Great. I hope we can follow up on that. I look forward to our institute administration helping and we have several of our translation centers, including SIGN and others, and many other entities, including WRCB and so on here. We hope they will all rise to this challenge. I think, Dr. Bhavi, you had a follow-up?
Just follow up on Dr. Pramesh. One of the things challenging in India, particularly from the clinical side, is the demand for clinical services is so high. Doctors have no time for research. This is a problem. In Chitra, we had our own hospital and a research wing, and I couldn't get the clinical faculty to come to research wing and spend time. So 2005, when I was in Utah, University of Utah at Salt Lake City, I found that they had a very interesting model where, I'm talking about doctors, the clinical faculty, there were two classes. One was called a clinical faculty who would do 70% clinical work and 30% clinical research. And then they had research faculty who would do 70% research and 30% clinical work so that they don't get dissociated from the clinical practice side, but these are the doctors who would interact with the engineers and all that. That was the model I suggested to our institute body, but it didn't get fine. I have not seen any other institute look at that. I think that's a very, very critical, important thing. Unless we find clinicians who have the spare time to really get involved in research. The other thing I remember, just one point, was a mechanical engineer who was working in the Department of Orthopedics in the Massachusetts Dental. In fact, he came back to India and he wanted to join IIT Madras. I was on the selection committee and he had some excellent 30 papers on some of the long-term failures of hip replacements and some of the issues. I have not seen an engineer working full-time in the clinical department and producing some excellent, but he had only 30 papers and IIT Madras said, no, doesn't have enough papers for appointment. In fact, I invited him to Chitra, but he didn't want to come to Trivandrum. But the other mindset that needs changing from particularly the clinical academic side, I think we really need to find ways where clinicians have the spare time from the clinical work to get involved in research. And unless administratively we provide this, because it also impinges on their assessments and promotions and so on, how do we do that? Thank you.
Yeah, in fact, to take this point ahead a little bit, we at IIT Bombay are examining this closely to see if there is some way we can affiliate more closely with medical education, even at the undergraduate level where our engineers are very well known. But that's a discussion for the future. We will certainly take all of your advice as we go ahead on that thread. But I'm waiting for a last word on this question before we take a couple of questions from the audience. Dr. Raman Ganakakit.
I agree with both of them when they talked about issues. Now I'm going to be a little blunt in what happens. When a person comes with a product, he tends to believe that this is the best product. Because in ICMR we wanted to promote, because you have PM who suddenly set the TB elimination target to 2025, and we had a huge push for everything that we did, even including COVID time. There are entrepreneurs who come. They believe they have done it rightly, but that's just a belief, because they have not validated it the way it has to be validated. You have to keep on sending them back that you do validation well. One thing which I realized was it's not only an issue of giving an advice. They don't know where to go in order to do this validation. You will find the same mentality that we have in most of the clinical trials that occur in India. You will find they tend to occur in such small places that you wonder where there is any kind of quality that is associated with it. And then you start questioning what are those findings. The same thing happens here. He would go to some person who he knows goes in the OPD and tells him, Acha, I want 100 samples and do it like that. It doesn't work. So one of the things which we did was I started mentoring institutions. Now suppose, because one of the needs of the country is antimicrobials, resistance is a major issue. Now if you want people to be assigned to different mentors, we could do both, supervise what's happening, and at the same time, they were able to produce a third party data, because most often people tend to say, I have done this, but there are no failures in it. If you actually try and validate those through any center, it doesn't match. That's one thing. Second thing, we have the brightest of the minds, no? Whether it is engineers or passionate people who want to do some kind of stuff for medical interventions, whichever way. But it is unfortunate that there is no industry-academia kind of a relationship, where more often than not, what happens is they prioritize something which is not a national priority. Then one starts worrying, and when you look at that individual, the way he speaks passionately, he has done good work, but what do I do with that product? No, that becomes a problem. That's second thing. Third thing, I think more than funding issues, that's whatever I have seen the country tends to show, no? More than getting funding to do research, what is most important is translation of this research, selling that patent, willingness of the partners, the other marketing person to actually adopt it. That is the greatest this thing. I really do not know what answers can we have in this particular issue. You will find he comes from NIV to sell their original tests to any pharma company. It used to take 3 to 4 years time. You have to fight hard. Companies also know, this is a good product, I can take. But their approach is not of innovation, but their approach is more of, is this going to get me money? If it does not, even if you have innovation, you will not be able to move forward. And this is coming from ICMR, which is a government agency. You have to almost force people, अच्छा ये आप करके बताइए. Nothing other than that. Like I was saying in the side talks, there were two major achievements that we had during my tenure. We developed a molecular test, point of care test for leptospira. We developed a point of care test for Nipah. These are the only tests which do not have US FDA approval, but our own CDSCO's approval. We directly licensed with the manufacturer at that time, and we provided that support, and they were very much needed kind of tests. But what is the tragedy? Is there a market for it? No. They produced it. But now to maintain that, how do you do that is another question. So post-research related, marketing related parts are also important.
Great, thank you. Thank you very much, Dr. Raman Ganga Kedkar. I think points have come up about funding, so I'm glad to see Mayur there, Mayur Sirdesai. So I guess we need to ensure that we are able to get, help us with issues of that sort as we go ahead to ensure that the long runway needed for many of these products is satisfied. And not only Mayur, we have several of our friends, colleagues, and alumni in that field, and we look forward to their support. So, Indranil, do we have time for questions, or should I close here? There is no time. Oh, that's the most unfortunate part. But well, we have to accede to the. Two questions. Okay, thank you, sir. All right, so we have kind permission to have a quick question or two. May I ask in the audience if we have? Is there somebody? Yes, please. Is there a roaming mic? Yeah.
Good afternoon. I'm Dr. Sayan Basu. I'm an ophthalmologist and cornea transplant surgeon. I work at L.V. Prasad Eye Institute. So we are also an academic research non-profit, and we have a GMP facility as well as operational since 2017. And currently we have manufacturing license for several biologicals that we are working on. So the question that I had was on the 10x. and I understand completely the concept that this is going to facilitate and make the discovery and taking it to clinical translation maybe 10 times quicker. But how do you control the costs? How do you make it 10 times cheaper? What are the strategies that the panel thinks could make it 10 times cheaper?
Okay, great. Thank you. Thank you for that input, and I'd love to connect with you later. Yeah, please. But yeah, let's. Who wants to take a stab at that? Yeah. So 10 times faster is certainly possible, right? 10 times faster is certainly possible, provided we have the necessary regulatory speed on the regulation side. I think the regulatory aspects are often time-consuming. The second aspect is getting in a clinical trial, getting the requisite number of patients sometimes is a real bottleneck for us, and I speak to that from personal experience of our product, which is going through multi-center trials. And in terms of cost, yeah, the issue there often is the cost of raw materials. And we really need to ramp up the availability of standardized raw materials, which may not be a medical grade, but we will get them tested through as a medical grade product for manufacture. I think this is an area where a lot of the other allied engineerings, for example, metal alloys are particularly useful, but very difficult to get internationally of a appropriate quality. That's where really our metallurgical and metal processing industry should contribute. Our polymer industry should contribute towards getting these exotic engineering polymers of appropriate quality. So this may take a little. This probably I think is the hardest thing to do. But that's a goal to strive for. So I don't think all aspects of the 10x are going to happen on day one. But I think that's where we are leading. Raj, would you like to? You coined the name and the phrase very appropriately, but
I think just not medical industry, but across industries, there are examples replete. I mean, the whole world is replete with examples of 10x or 15x. It's just that if you try to get a 15% or 20 or 30% improvement, you try to fiddle around with efficiency and effectiveness and so on and so forth. You'll never get to even 2x. But the moment you say, let me go back to the drawing table and see what do I need to do. Now, I don't want to disclose some of your secrets, but one of the products he's making, you'll be surprised the raw material cost is less than 5%. Okay? And nobody else in the world has got anything which is even close to 50%. Okay? Because it requires breakthrough thinking. Unless you get out of the kind of thinking you got in the existing system and say, I'm going to figure out how to make it much cheaper and think of a new way of doing it, you're never going to get 10x. So the whole idea is breakthrough. And breakthrough happens not by thinking along conventional lines. So the answer to your question is, if you're looking for conventional solutions, you're not going to get to anywhere. But across industry, if only we had the time, we could give specific examples of more than 10x. Even Professor Rahul Purva's CAR-T cell, which was inaugurated a few days ago, was at least 12x. Somebody mentioned 10x, but at least 12x. So there are any number of examples, but you have to be less lazy. If you want easy solutions, it's not there. You've got to look for breakthrough, and that requires guts. If you are only interested in producing a scientific paper, it's very easy to say you cannot get 10x. But if you think from the customer side back and say, what is the need and what can change the need, move the needle for the world, there's huge possibilities.
Thank you, Raj, for that. And I think on that note, we should. On that note, we can close this panel discussion. I should express my heartfelt thanks to the luminaries on the stage here to have graced the occasion, given us talks and participated in this. And we look forward to your continued support in various ways as this facility goes ahead and fulfilling the dreams not only of the donor, but also the flame he has kindled in many hearts by the figure that he quoted earlier. Yeah, thank you very much.
Thank you.
Yeah.
Can I request you all to wait?
So thank you all. May I request Professor Goody to come on stage and felicitate the panelist and the moderator.
Take one group shot. Oh, thank you. Take one group shot. Panel shot. Thank you. Thank you all.
नाही नाही आता बरं झालं तिथं खरं सांगायचं होतं मला स्टेल्थ टेक्नॉलॉजी इथं यायला पाहिजे मी लपलो.
As we approach the conclusion of this event, I request Professor Ravindra D. Goody, Dean Alumni and Corporate Relation, to please deliver the vote of thanks.
Thank you. Good afternoon, and make it quick. Vote of thanks is usually for a closure, but this is just the beginning of an exciting journey going ahead. It's really my pleasant responsibility to do this, I would say, a vote of gratitude on this very momentous occasion, the inauguration of the 10X GMP facility. I think it's more of a physical space. It's just an ongoing commitment of nurturing innovation and creating those products for societal impact, which is what has been the Institute's drive, and in the bio area it's been very nicely facilitated by Raj. So I'd like to begin by first saying a big thank you to Raj for his exceptional philanthropy, establishing this 10X GMP facility. Yeah, I think it's not only an outstanding generosity, it's an exemplary leadership, I would say, in every step as this GMP facility was brought up. A lot of, I would say, visionary guidance driven by his passion that has really set the stage for this transformative era at IIT Bombay. Of course, we look forward to collaborating with you, receiving your generous advice as we go ahead on this in the coming years. I would like to thank our chief guest, Dr. Pramesh. I think your presence truly enriched and elevated today's inauguration. I was telling Dr. Pramesh that I'll go back and revisit the YouTube recording just to look at various initiatives that he proposed, which are opportunities and challenges in the healthcare area for the country. Thank you, Dr. Pramesh, for your presence here. I'd like to thank our guest of honor, Dr. Raman Gangakhedkar. I think your storytelling enriched the occasion very well, and of course your advisories later on in the panel discussion were indeed very, very helpful, very inspiring to all of us. Thank you for taking your time to come from Pune and being present with us. Really enriching. Dr. Bhuvaneshwar, thank you for being our guest speaker today. Again, your experiences, your journey throughout this biomedical innovation, certainly very inspiring, and we will derive a lot of guidance from that. May I request our alumnus Ajit Javale to kindly stand up. I think he does require a very standing innovation here. You know, we came in contact during COVID when Hostel 5, he belongs to Hostel 5, and his involvement in the renovation of Hostel 5, actually the activity started much before COVID, made us realize that, of course, his strong love and attachment to the institute, but more importantly, the tact. We learned a lot about how tact, diplomacy, and getting things done in a project management mode, dealing with various stakeholders and being able to convince them, cajole them, coax them, get them along towards the intended objective. It's been a learning for me when I joined the online meetings every Saturday morning. Really thank you, Ajit, for the tremendous services, not only on the GMP, but also on several other initiatives with the Institute. You have a very rich experience, and the Institute will look forward to continuing to benefit from your association. Thank you so much for that. I think one of my hostel mates and of course another very accomplished alumni, Dr. Dhananjay Patankar, he is not here, he's on travel, but I would like to acknowledge his help very strongly in several ways through the GMP journey. He helped us with several aspects of the vetting the designs, as well as providing various advice on quality assurance. Thank you, Dr. Dhananjay, it's on record online. Of course, a big round of applause, Professor Jayesh Bellari, the silent mover in the background. I think he did an exceptional job in moderating this panel discussion with several august and eminent panelists, Dr. Shailesh, Dr. Pramesh, Dr. Ganga Kedkar, and Dr. Bhuvaneshwar. Thank you so much for the thought-provoking discussions. Really. So I would also like to take a moment to thank many of the associates who worked tirelessly behind the sessions. Firstly, it's my set of apologies, but also sincere appreciations to the entire biosciences and bioengineering fraternity, the faculty, the researchers, the students, because right in the midst of very exciting research that they were doing, this construction activity, accommodating the inconveniences associated with this activity and cooperating with all of the schedules. I think a big round of applause to all of the biosciences and bioengineering headed by, of course, Professor Balaji, formerly Professor Rohit, Professor Ambarish Kumar, who was very actively involved, Professor Shamik Sen, Professor Prakriti Tayalia, all of them, including the entire biosciences fraternity, would like to really appreciate them accommodating all of these inconveniences that happened in the interim. Our contractor, MK Precision, has been truly a partner in this entire process of establishing the GMP. Our sincere thanks to them for patiently accommodating our, we had a lot of elaborate procurement procedures, and the time it took, the delays they involved, I think they very, very silently accommodated that. Thank you so much for that. Also, the Team Tech Solutions Projects Private Limited, Team Excel Constructions also played a key role in the project implementation. And of course, to Yatin Shah, been involved in several aspects of this project. Sincere thanks. A special mention must be made of my colleague, Dean IPS, Professor Tom Matthew and his office. Of course, the EMD division and the State Office for playing the perfect partners in our journey when we had to put all of these facilities together. They really helped us in various aspects of the electrical and mechanical aspects, the construction aspects and the like. We would also like to acknowledge support from the Institute of Eminence Cell, the IIT Bombay Heritage Foundation, eClinical Works, L&T Technology Services, SKF India Private Limited, Lavinstar Logistics and Castle Ship. They provided us support in various forms, and we sincerely acknowledge all of this help. The Public Health Office, PHO, and the CDEEP who have been recording this, thanks very much for your time and for your support in this matter. There is one individual who was mentioned in passing, but I would like to request that Ms. Nidhi Mathur kindly stand up. She deserves a standing ovation. She is heading the PM team, which stands for Project Management, but she has also been the prime mover, balancing various stakeholders and getting all of this activity in place. Thank you so much, Nidhi, for and your entire team, I would say, for the exceptional work. Again, my heartfelt appreciations to the dedicated members of the DRF, marketing team, the major donor team and the like, who have supported us with their significant enthusiasm, creativity in putting all of this event together, what we are seeing today as the event. And then finally, on my own personal behalf, I think would like to, this is, as I said, just the beginning of a very exciting journey the institute had, recognizing the significant forays in the healthcare area. In fact, if you see Sign, about 40 to 50% of the companies in Sign that are being incubated are in the healthcare area. And the BSB department, as well as several other departments like chemical engineering, physics, materials, all of these continue to be a hotbed of innovations in the healthcare and biomed area. I would certainly look forward to continuing collaborations with the broader fraternity of researchers in the healthcare area, the hospitals, the other NGOs and several other entities the pharma industry, the corporates who work in the healthcare area. We look forward to our continued collaboration in our exciting journey, both from the perspectives of being enablers, but also the users of this GMP facility. And really, I think we are at a very momentous point. Going forward, we continue to look forward to stay in touch with all of you. So thank you very much. I know I'm standing between you and lunch, but let's continue the conversations over lunch. Thank you.